Good morning, everyone. My name is Naman Jain, and today I'll be presenting my presentation on identification of transcriptomic signatures for tumor staging and classification. So, my first slide is what is bioinformatics. Bioinformatics is the use of computing and analyzing tools for the interpretation of biological data. In modern biology and medicines, bioinformatics is a critical for data and management. Computer softwares packages like Blast, Ensemble, which rely on internet access, are part of bioinformatics toolkit. Transcriptomics. Transcriptomic technologies are methods for studying an organism's transcriptome, which is the total number of RNA transcripts as organism's information content is stored in the genome's DNA, and expressing through transcriptomics, non-coded RNAs perform additional roles, whereas mRNA acts as a temporary intermediate molecule in the information network. The entire transcripts present in a cell are captured in a transcriptome, and which is snapshot in time. Transcriptomics in cancer. Transcriptomics is very useful method for studying cancer causes and identification biomarkers. It includes assessing alternatives, slicing, and polydetonation detection, fusion, transcriptome, exploring non-coded RNAs, transcriptomes, and annotations, and discovering new transcripts. Large-scale gene expressions profiling in new possible because of recent improvements in high through output technologies. So the types of tumor we are dealing with are basal-like, normal-like, luminal, and claudin-low. So basal-like breast cancer, BLBC, is a genetic subtype of breast cancer that is characterized by a large cluster of genes produced by epithelial cells in the adult mammary gland, basal or outside layer. Because the tumor are common in young women and typically relapses quickly, BLBC is a serious critical problem. So the next is normal breast cancer cell. Breast cancer starts in the tissue of breast. Breast cancer develops when cells of breast mutate, alter, and expand into uncontrollably, and it results in tumor. Mass of tissue uh, like cyst is formed, and then tumor is formed. So breast cancer are malignant, and it is surrounded by breasts. The third is luminal breast cancer. Around two-third of breast cancer of estrogen receptors ER positive or luminal tumor. Luminal breast cancer has a wide range of histogenic gene expression profiles and mutational patterns, as well as wide range of clinical outcomes and responses in systematic therapy. And the fourth is Claudin-Low. Claudin-Low breast cancer is a molecular form of breast cancer that was discovered using gene expression profiling, and has been linked to a poor prognosis. Claudin-Low tumor have long been through favor of triple negative phenotype, yet only a small percentage of triple negative breast cancer tumor are Claudin-Low. So here we'll be comparing two types of cancers. So we can look here. This is the normal like cell, and this is the Claudin-Low cancer cell. So we will consider this as array 1, and we will be consider this as array 2. So we have to make a comparison of all the cancer types like luminal normal, luminal basal, luminal Claudin-Low, normal basal, normal Claudin-Low, and basal Claudin-Low. From that, we can find out the FDR value, the P value, and the fold. And through every detail, we can find the genes related to the group. So how do we find p-value in this? So we can find p-value by using equal to t dot test, start of array 1, end of array 1. Let me show you. Start of array 1 is normal like and normal like here, and this is the end. And start of array 2 and end of array 2. This is start of array 2 and this is the end of array 2, comma 2, comma 3. This will give us the p-value. Then we'll add count in our Excel sheet, which is very easy. And then we will add FDR value for that column of p-value, as like the column which has the p-value multiply by maximum of count, count is to count, divided by third column of count. And for fold, we can use the formula average array 1 array 2. So this is the average between array 1 array 2 and minus average of array 2 array minus array 1 and array 2. So from this we can find out the formula of fold, and from that we can find out the top 10 upregulated genes. So for luminal and luminal normal, we have these: luminal claudin these, luminal basal these, normal basal, normal claudin, and basal claudin. And then same goes for downregulated. So these are the genes responsible for the least amount of cancer regulators. So we have to make this. From this we can find out that these these genes are regulated by luminal genes. These two and this one is regulated by the normal gene, and basal claudin and these two are regulated by the basal genes, and these claudin low genes are the ones who are the who are responsible for the down regulated genes. And so I'll be ending my presentation here. Thank you.